have Anna Cates. She is our state soil health specialist, representing the Minnesota Office of Soil Health, and is also part of the University of Minnesota Extension team. So Anna, take us home.
All right, I thought we were just going to watch YouTube videos there for a minute while Tim had that up, but I guess I do have to actually give my talk. I'll do my best. I'm acknowledging a couple of collaborators on there, and I'll try to call out a few more people when they are relevant. So as he said, I'm from the Minnesota Office for Soil Health. This is a state office that mostly consists of myself and Marcel Lewandowski. We have funding from the Board of Soil and Water Resources through the Clean Water Legacy Amendment. And our mission is to increase adoption of soil health practices across the state for the benefit of water quality. This has been in place about 6 years. I'm a soil scientist. I did my training in soils and agronomy over in Wisconsin and came over here after my PhD. So I do research and extension and kind of network building around these ideas of soil health in Minnesota. Okay, I'm going to show the summary slide at the beginning and at the end, and you'll kind of see the sort of hand-wavy conclusions are what I have to offer on this topic of predicting corn nitrogen needs with biological tests. So I'm going to talk a little bit about what we know about nitrogen supply to corn. A few people have talked about this already today, so that makes my job easy. I'm going to talk a little bit about what measurements are out there and available to predict nitrogen supply. And I'm going to talk a little bit about what that might look like that might be different in a soil health system. The QR code that's up there is for the series of podcasts that my colleague Brad has been doing based on his Nitrogen Smart curriculum, but they're very digestible in podcast form, cover a lot of this information in that format if you like listening to stuff. Okay, so this is the soil food web that I like to show. And I don't think we did the thing where I get to have a pointer over here, did we? I might actually stop and do that. You want to help me, Tim? Where is it? Okay. There we go. That works. Okay, thank you, Tim. Okay, so I wanted to be able to point to this because I'm going to show this a few times in a few different forms, and it's a little bit complicated, right? So this is a little diagram that shows the flow of energy through the soil food web. The arrows actually mostly refer to where carbon is going, where food is going in the system. Things are eating plants, other things are eating the things that are eating plants. It's just like it would be in a system you might learn about in a third grade biology lesson with grass and deer and wolves, right? But instead of that, we have microbes, we have worms, and we have, what passes for predators in the soil environment, which are things like collembola and mites and centipedes and nematodes. So the thing I don't usually put in on this slide is that every single one of those arrows has nitrogen attached to it also, right? So where carbon goes, where organic carbon goes, organic nitrogen goes too. So when we think about how to understand where nitrogen is flowing in the soil, we actually have a lot of different movements that we could be tracking, which is kind of going to be my excuse for not knowing all the answers here. Okay, I think this has a lot of there we go. I got all the animations done. This is just the nitrogen cycle, which I think we've seen a couple of times today. But I want to point out where we are in this nitrogen cycle. I'm really talking about this pool of organic nitrogen. I'm going to talk a little bit about the transformation of that organic nitrogen into inorganic nitrogen, which has been mentioned a couple of times today. Interestingly, organic nitrogen is labeled here as a stable nitrogen pool, which I would kind of argue that it's not. But my master's student made this a few years ago, and at the very least, it's legible and pretty in its presentation. So we'll try for accurate or perfectly accurate next time. Okay, so I pointed out how nitrogen is moving through the system, but I didn't point out in the last time I showed this how nitrogen is coming in and leaving the system, right? So nitrogen can be lost during these transformations from one organism to the next. In particular, I'll point to this big arrow, the fact that nitrogen can be lost when a larger organism eats a smaller organism. That's because these smaller organisms are richer in nitrogen than the consumer needs. And I'll show a little diagram of that to help. So this is essentially where nitrogen is released or mineralized in the system a lot of times is through this active predation that essentially leaves some leftover nitrogen. Another thing I'm going to show here is the immobilization step, that sometimes you're going to have nitrogen coming in from the soil and being immobilized in microbial bodies. And this was alluded to a couple of times today. People talked about how the carbon to nitrogen ratio, when it is very high, you're going to have nitrogen pulled in from the soil. So that's what this particular arrow is supposed to be showing. Otherwise, I'm just showing that it's a leaky system, that if you have a microbial, a bacterial body die in the soil, and then another organism comes along to use that, they probably won't be likely to get every single cell before some of it is transformed into soluble nitrogen that is going to leave the system. So that's what those little nitrogens represent. So this is kind of what we're dealing with. When we had people earlier today talk about how, how much of that fertilizer nitrogen is in the organic form and how much of your corn crop is, or how much of your corn nitrogen is coming from that soil nitrogen, it's coming from all these incidental releases of nitrogen along all these transformations. So you have to remember that essentially none of these processes are designed to feed nitrogen to your corn crop. They're designed to help, the next organism up the food chain survive. And so when we think about how to measure how much nitrogen incidentally gets to your corn crop, I want you to remember that's kind of an accident every time. It's not really anything that's purposeful or deliberate along the way. Okay, so let's talk a little bit about the carbon and nitrogen processes that drive kind of major inputs and outputs to this organic nitrogen pool. I'm going to start with this one, the immobilization one. So this is showing that if you have a cover crop, say, with a carbon to nitrogen ratio of about 40:1, this would be any plant that is kind of close to that reproductive stage, that senescence. Sometimes your corn residue would be similar to that 40:1 level. Really anything that looks pretty brown might be around there. Then you have a bacteria which might be, hungry for corn residue, but its carbon to nitrogen ratio is 5:1, right? So if they are going to consume the rye carbon or make more biomass, right, and bacteria, are multiplying and making more little bacterial bodies, they'll take 5 of those carbon right off the bat and that first nitrogen, and they'll make another bacteria. But then if they want to keep consuming that, they have to take soil nitrogen for all the rest of the reproduction they might want to do. And to some extent, they might not do this all at once. They might not take all of those, 6 or 8 more nitrogens that they need to make more bacterial bodies. Instead, it might just go slowly, right? That is really probably more likely what's going to happen. But they're going to take some soil nitrogen along the way. This is just showing that microbes prefer that low carbon and nitrogen ratio. I don't think I'm going to stick around here. But now I want to talk about the other part of the process. This is how you might get mineralization after that immobilization process. So this is where, I'll skip ahead actually and then come back to this. This arrow I'm trying to depict here is when you have something larger eating a microbe, how you end up with extra nitrogen in the system. Okay, so my predator in this case is a bacteria-feeding nematode, and the premise is kind of the same. The carbon to nitrogen ratio of that nematode is 10:1, whereas the bacteria C to N ratio is 5:1. So they consume 2 bacteria, they get enough nitrogen from just one of those, they keep all the carbon, and they're excreting part of the nitrogen, and that's going to be a solution available. So this, some people say this accounts for, just this act of predation by things like nematodes and protozoa. This is accounting for a huge proportion of that soil mineralizing N. It's not something we measure very well, which I'm going to talk about a little bit more later. Okay. So I think I've accounted for kind of the major points of entry here in terms of soil nitrogen becoming in this this organic, supposedly immobile form. And then when it comes out is largely over here or any of these leaky processes as things are transformed. Okay, so these are some of the measurements that we can do in the soil that might help us predict how much nitrogen is going to be part of this arrow go into crop uptake. One is this question of just the two pools, the organic nitrogen and the inorganic nitrogen. And I'm going to show a few inorganic nitrogen slides because that really is the gold standard. It's easy to measure. We have a lot of data relating inorganic nitrogen to crop outcomes. Organic nitrogen is another potential pool. I'm not going to talk a ton about that, but keep in mind when we're thinking about organic nitrogen pools, we're essentially taking all of this together and saying, you know, how much nitrogen is there in it? It doesn't tell us very much about how fast this is going to happen, which might be the process of greatest interest here. And so then I am going to talk a little bit about metrics that assess processing, how fast microbes can transform nitrogen. So you've seen a few figures like this today. I just pulled some off of the internet, trying to show the relationship between inorganic nitrogen and yield, or relative yield in the case of these two. These are from Ontario, but we've seen some from Minnesota here today too. So that inorganic nitrogen is usually in the form of a pre-plant nitrogen test. In Minnesota, you know, we recommend this at specific places and times. You know, definitely going to be useful over in this quadrant half of the state, and then over here we have more of a decision tree as to when that pre-plant nitrogen test is going to be most useful, and it has to do with when we think there's going to be significant inorganic nitrogen available that's going to actually change how much nitrogen your crop needs later. The other thing we think about with inorganic nitrogen is that pre-side dress nitrogen test, and I'm showing this kind of classic study on the pre-side dress nitrogen test from Wisconsin that shows that there's really a difference, especially in the soils on the right with high yield potential, where when they used the pre-side dress nitrate test, they were able to correctly apply, you know, more in proportion to those that were over-applied or under-applied. Whereas if they did a standard one on those high yield tests, they over-applied quite a bit. So it has a lot to do with what you think your soil has in it to begin with, of course. Okay, here's an effort by one of Fabian's students. I think he alluded to this a little bit earlier, but I think it's probably the most comprehensive effort we've had so far in Minnesota to add to our inorganic nitrogen tests to predict corn outcomes. So this graph compares nitrogen. Nitrogen in the soil is on our x-axis to our relative yield on our y-axis. And the first one is just the correlation between the soil nitrogen and the PPNT. And then over here, they added that potentially mineralizable nitrogen test. So potentially mineralizable nitrogen is when you take your soil and you put it in a jar and you keep it in anaerobic conditions to see how much microbes are going to process organic nitrogen into inorganic nitrogen. So it's a metric, right? So they took some samples at pre-plant, and they were able to add that to the PPNT and see if that explained relative yield more. They took samples at V5, thinking that might be a better indication of your microbes' ability to improve, or your microbes' ability to supply nitrogen. Didn't really improve things. And then we have that, this column is the correlations when they took samples in zero nitrogen plots, and over here, samples in plots with nitrogen applied. So this has data from all over the Midwest, and PPNT is still kind of our best indicator. So in terms of what we have so far, I would say that the processing indicator hasn't been especially good at using that prediction of how much microbes will transform nitrogen and translating that to an actual yield outcome. Okay, I'm going to talk a little bit about organic matter. So just organic matter actually does an okay job of predicting yield, especially if you're on low organic matter soils. So these figures both show organic matter relative to some metric of yield. This is a greenhouse study, so it just has above ground biomass. This is study from field sites across Wisconsin and Minnesota. I believe they're Discovery Farm sites. And it has yield relative to soil organic matter percentage for silage and grain corn. And in both cases, you see kind of a plateau in there. You know, yield isn't really continuing to increase after around 2.5% organic matter. And over here, it's the same. So you can just say, well, like my straight organic matter, if I'm over 2%, isn't going to say much about what kind of yield I'm going to get. And I saw at least one other presenter today, I think it was Fabian again, talk about how your nitrogen is part of the picture, but when you have other yield limiting conditions, then your organic matter or your organic nitrogen isn't likely to be a good predictor. I have some really smart colleagues in the soil health world who have done really complicated models where they measured a lot of different soil organic matter pools and tried to figure out which were the best predictors of yield. So this one picked out, latitude, so where you are. It also picked out things like the permanganate oxidizable carbon, phosphorus. There's a couple other soil health parameters on here, organic nitrogen, carbon, and they all together could better predict crop yield. This figure on the right shows a similar thing where you can take your soil health indicators, including carbon, soil protein, and permanganate oxidizable carbon, and predict relative fertilization and relative yield. Great. But for something to actually be helpful, I think, in terms of making in-season fertilizer decision, it's something it has to be simple and quick. You have to be able to take a sample, in advance. So you have to have your plan that's made at this time of year. You have to be taking your sample in the spring or even in the fall. You have to be able to adjust that in-season nitrogen, and then it has to actually be, profitable in the end. Yeah, it reminds me a little bit of the South Park meme where you're like, is this actually leading to profit, or are we just pretending there's profit at the other end of it? Um, so I just want to point out though that we have a lot of really cool ways of looking at the soil biology and soil organic matter, but what we are struggling with in my world is a test or a metric suite of tests that will fit within the agronomic timeline of you had to make the plan way in advance because you made your fertilizer contracts way in advance. You have to be able to take that sample and get it analyzed and have to be able to make that in-season adjustment, all with a level of return that makes it worth all this trouble you've just gone to. So I don't know if we're going to get there, to be perfectly frank, but those are the standards that I am putting these metrics to when I think about the data out there. We might have really good predictability if we do these 17 different things, but it's not something we're going to be able to get done in April and then adjust in season application rates. Even just a few of these, might cost $50 a sample or something like that. Okay, so there are some things that show promise. I am not gonna, quit my job and go, I don't know, work at Target or something yet. But so I'm gonna show some things that do show some promise that are because they are kind of within my realm of they fit my sieve, I guess, for being reasonable. So this test I'm going to talk about is what sometimes people call a carbon dioxide burst. It's under the hood of the Haney test as soil respiration. Generally, you put a little bit of soil in a cup in a lab, keep it at room temperature and at a predictable moisture level for just 24 hours and see how much carbon dioxide comes out. So this is really specifically getting at how active are your microbes. It's not perfect. It's going to be really correlated with organic matter, but it should get a little bit at some level of microbial fitness. This is some data that Marshall McDaniel put together where he normalized CO2 bursts. So 1 represents, the best CO2 burst score in that study. 0 would be the worst. And then he has some studies with economic optimum nitrogen rate correlated with the CO2 burst. That's 2 different studies here in 2 different parts of the US. And he shows a pretty good relationship. So the concept here would be that as your CO2 burst gets higher, your microbes have more fitness and they might be able to more effectively deliver that nitrogen over the course of time. Maybe it means you have a larger population. Maybe they're just very efficient. We don't really know from this test, but we know that there's something different about those. And the other way Marshall looked at this data that I liked was he pointed out soil A, which essentially doesn't need any nitrogen, has a really high yield regardless of fertilizer application, and it has a really high CO2 burst test, right? Soil B, it's very responsive to nitrogen, and the CO2 burst test is lower, right? So this is how we hope it might be effective, is if you have a high CO2 burst test, we know that you don't need as much nitrogen. We don't have really good data on this in Minnesota, so I piggybacked on some stuff that Jeff Vetch and the MDA were running in southeast Minnesota. So this is on-farm trials that they did over several years. They are ongoing. They were in farmers' fields, but they, ran 4 replicated strips of different nitrogen rates. Um, there are a couple of different sub-treatments in terms of nitrogen timing that I'm not going to talk about today, but they were able, from their trials, to calculate an MRTN. And then I ran the CO2 burst in the 0 nitrogen plots on the premise that what we're interested in is what the soil can deliver by itself without some of the stimulation of microbes that Julie Howe talked about. So again, just to remind you, these relationships should be negative. Our idea is that, like, if these guys are really kicking butt out there, that we should need less nitrogen applied in the spring. This is the ideal relationship. What we actually see is no or positive relationship to that maximum return to nitrogen. So it was not, what we would expect from the biological theory. The other thing you might expect is that these especially effective microbes might just by themselves deliver more nitrogen to a 0 nitrogen plot. So we looked at that too. And the 0 nitrogen plot here is on the bottom. And then the yield at MRTN, so that sufficient fertilization rate, is on the top. So in this case, we would expect a little bit of a positive relationship, but we're not really seeing that either. In fact, some cases where we have really pretty high CO2 burst tests, we have actually lower yield at that MRTN. So I think, this is again echoing what some other people have talked about today, is that in Minnesota, I think some of these sites are struggling with drainage. Some of these years have been wet. We've needed a lot of nitrogen to get to good yields in these systems. So their CO2 burst is kind of not the most important thing that's going on for corn production in these sites. And I think we need to sort of continue to look for better sites. So just one little cartoon here where they talk about a scientist being asked to predict the future. And he says, "You are equally likely to experience opposite results today," which I think was something Kenny said this morning. And so I definitely, when I look at these results, I have a little moment. Sometimes science is so difficult, it makes me sad. But I do want to talk about what I think are worthwhile ways forward to work ourselves a little bit out of this conundrum. So this is some more work from Marshall McDaniel, who I think has been at this meeting. I really admire his approach of integrating soil health with some practical agronomics. Um, he was looking at the difference in soil health between sites that were responsive to nitrogen, that's this RN category, high yielding non-responsive, and low yielding non-responsive. And he was pointing out that sites that are responsive to nitrogen generally have low nitrogen. So you put nitrogen on and they grow more corn. Sites that are high yielding but not responsive tend to have high results, but it's these low yielding ones that seem to be kind of all over the map. So he's saying this is kind of the area where we could be most effective. First of all, we don't want to apply extra fertilizer on these. So if we could identify them, that would be a good economic savings. And then second of all, just to better understand what is going on there, maybe it's agronomic, but maybe there is something a soil test can tell us. So he used a bunch of different soil tests and was able to kind of tease out which soil nitrogen tests were most responsive or most related to what he used was an agronomically optimum nitrogen rate instead of that economically optimum nitrogen rate that we've seen in other studies, but those tend to be fairly aligned. So he was able to say, like, okay, our responsive sites tend to, be a little different than our non-responsive high yielding and low yielding sites. So I think that there's something to be said here, and this might be something that we could apply a little bit more in our Minnesota data sets and try to dig into which sites deserve a little bit more attention. Where can we actually, where are we on this curve? Are there sites where we can really boost our yield versus are there sites where we can kind of just back off and not put a lot of inputs in? The other thing I'm interested in is this question of whether, when that soil nitrogen is released and who is responsible for the releasing. So I've pulled this off of a kid's soil site, but it was really just exactly what I wanted because I want to understand a little bit more about what those predators are up to and how active they are and how responsible they are for releasing nitrogen in the soil system. Unfortunately, I don't get to do any safaris, but I'll maybe do some work figuring out how to count, predatory nematodes out there. That's a more accessible thing for a soil scientist. And this is just, there's pretty limited data on this, but there's some data that says, okay, if you have more invertebrates in the system, you might be able to grow more biomass. And then the other thing I'm really curious about is because I'm part of a position and a system where we're interested in promoting soil health systems for other reasons besides corn nitrogen supply. I'm interested in whether a soil health system where we reduce tillage and we include cover crops, is there going to be a difference in the nitrogen supply in those systems? So the main reason, right, would be that we have more roots in the ground in these systems, right? They're supposed to stimulate the biology by leaving the residue on the surface, having a root in the ground for more of the year, which is going to boost that biology. But the other reason they might change how soil nitrogen is supplied is because of the soil structure here. So these are some videos I took on the St. Paul campus showing after a little rain demonstration, we used this little canister to rain on the soil surface. Pretty gentle rain. We rained for about half an hour, just a little sprinkle. And in the system that had had a lot of tillage, this is what it looked like after half an hour of rain. In the system where we had about a full year of rye growing, it looked like that. And I think that the benefits to this soil over this soil are obvious in logistical concerns, in our ability to keep water around, like Jeff Strock talked about earlier today. But they are also going to have an impact on how nutrients are delivered to your crop, right? There's just not a lot of movement in this soil. Once you get a little rain on that, it's just nothing else is going to happen. So these are the soil health principles. Uh, I was listening to Brad's talk, and I know you shouldn't do systems research because you can never really tell what is the thing that is causing the effect that you see. But I am interested in soil health systems as a full farm management approach because I do think that these principles kind of go together. So these are the principles the NRCS promotes. I think that Julie did a good job of going over most of them. I do keep integrating livestock on the list because manure is such a great nutrient source in the system, and it can kind of make the other practices pay. So in a row crop, Minnesota corn soybean system, that really looks like reducing tillage and adding cover crops, right? When you're in this kind of system, though, one question that is out there is, when does the change become apparent? So the study I'm just starting this year, I'm going to talk about for a minute, really tries to get at that. This is a figure I pulled that just talks about first the rate of change in soil carbon, and the colors represent different depths of the soil after no-till adoption. And then this is the actual change in soil carbon after no-till adoption. So they talk about like a dip early on, but then it peaks later, but then maybe it stabilizes after 50 years. And these are, I hope, obviously modeled results. We don't have a lot of actual tracking of this kind of data over time. But there's a lot of question as to whether a system is in equilibrium or is mature or what have you. So I think that that question deserves some attention in terms of how these soil health systems function on the landscape. So some of my next steps, after I got done, crying about how hard science is, then these are some of my next steps. We're going to stick with that Southeast Minnesota site. I think it might be interesting to categorize our site years by whether or not they're responsive to nitrogen and see if that changes how they fall out. And then we might consider some of the other nitrogen tests that Iowa study found effective. And then we're going to just look at other sampling depths because we do have, I think I showed you just 0 to 12-inch samples, but we do have 0 to 6-inch samples also. And then I'm going to talk a little bit about this new study where I'm going to get a little bit at that question of, what is the long-term effect? So we're doing these nitrogen rate trials. This is funded by the corn growers. Fabian's a collaborator on this, and Morganka Day from MSU Mankato. So our idea is to say, is there a difference between the long-term soil health site, the short-term soil health site, and the conventional site in their nitrogen response? So what I'm showing here is a soil map of a cooperator's field, and then the map of the little plots that we'll put on top of that. We're going to measure things, similar things to what I've showed before. So I'm Not sure it'll be perfect, but I do think that some of these we have reason to believe that they could explain the changes that you hear people talk about when they talk about a change to a soil health system. So I hope to do the soil respiration and microbial biomass. I'd like to get a nematode community analysis. Nematodes are unique because they live at essentially every trophic level. They could be fungal feeders, bacterial feeders. They can eat each other. They can eat, I don't even know what all they eat. But essentially you can say are there more predators? Is there a good amount of prey for a predatory nematode? And that can tell you something about how well, how well the whole food web is stable. And then we will probably keep that potentially mineralizable nitrogen one in there, even though other research has not shown it to be perfect. It's not hard to do, and it keeps us kind of able to compare our results with other people. Okay, I might be finishing a couple minutes early here, but I want to show you my summary again. We do know quite a bit about how the nitrogen is moving through the soil system. We don't have a perfect measurement for how to predict that nitrogen supply to corn. There's some reason to believe that a system which reduces tillage and includes cover crops would stimulate biology enough to change delivery of nitrogen. I do feel like it's important to say that uh, there's only so much nitrogen in the soil. It can't increase that delivery of nitrogen infinitely. It could change the timing of it, make it more accessible. It could make it happen at the plant's roots, maybe if you have a more active root zone. There are ways that the mode of delivery could be changed, but there is it's not making that much more nitrogen unless you have a very healthy clover cover crop going on in the fall, which not very many of our corn and bean farmers are able to do in Minnesota. So there's some potential for this, but it's going to require some digging to really understand that nitrogen delivery that could be changed in the soil health system. Okay, I'm going to wrap up there. These are some of my collaborators in my lab group, including my students took a selfie right outside this building a couple last week. So they almost got it in front of their posters, but not quite. Yeah. So I'm going to answer your question a little bit indirectly. Soil health can mean a lot of different things. I would say if you're looking for, a little biomass in the system and definitely getting your ground cover, I would be really happy with about 500 pounds an acre of cover crop, which I think Jeff showed some pictures that were in that range. But, you're going to see some bare ground in between it, but you also you're going to drive by the field and think it's green, right? That's important. Um, when we think about the biological effect, those roots are definitely always pumping carbon into the soil and supporting microbes around them. But what I think I wanted to highlight here is that the cover crop is holding the soil in place. So this is wind erosion data from Polk County from 2022-23 winter. And you can really see the dramatic effect that the rye cover crop had in reducing soil movement. And if your soil is blowing across the landscape, it is not healthy. So it doesn't take very much cover crop to keep your soil in place. This was definitely less than 500 pounds an acre of biomass. I mean, Polk County, Minnesota. So I think almost all of us in the room are south of there. So it doesn't take a lot to get at least that soil health benefit of holding your soil where you want it to stay. Yeah, we're going to take some data at the research and outreach centers comparing overwintering and winter-killed cover crops in terms of water erosion benefit as well as nitrogen uptake for the next couple of years. I have a master's student funded by the Minnesota NRCS working on that because I do think that for different resource concerns, we're going to have different cover crop needs. And so if you might get enough ground cover just from the residue of a really healthy oat crop. We definitely did in Wasika this year, but Jeff showed some years where we did not get that. So I think it's going to depend a lot on what your goals are. For stopping erosion, I think you might be able to get away with a pretty minimal amount. But I think for an overwintering cover crop, as many people showed today, is critical for taking up the nitrate and getting to the water quality goals that a lot of us have. Oh, research been done on the effectiveness of rye on control of soybean cyst nematode. Um, no, I hear anecdotes about this, but I have not seen much research done about it. I think it would be really interesting. One thing I know, my friend says that when he sees the soil blowing across the landscape, he doesn't just cry about the lost organic matter and degradation of soil quality. He sees soybean cyst nematode blowing across the landscape. So certainly rye could control the movement of SCN.
All right. Well, let's give Anna a round of applause.